Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to today's webinar titled RWE Reality Check, Routine Patient Health Records and Regulatory Requirements. In this webinar, these four experts will explore the key differences between clinical trial data and real-world data, and how to bridge this gap to ensure robust and reliable RWE that meets the regulatory grade threshold. And now I'm very pleased to welcome our moderator, Dr. Liz Mearns. She is the Director of Business Development for Real World Data and Evidence at Cardinal Health. Liz, welcome to the floor, and please take it away whenever you're ready.
Thanks, Sarah. Hi, everybody, and welcome. My name is Liz. I am the Director of Business Development in our Real World Data and Evidence group at Cardinal Health. We have an excellent webinar for you today. The agenda is here on the slide for you to see. We'll be talking about a reality check for real-world evidence. As we all know, data plays a crucial role in shaping healthcare decisions, regulatory approvals, and patient outcomes. But it's in the way that we collect it, interpret it, and apply it that can vary significantly between randomized controlled trials and real-world evidence. There's a big difference in the way the data looks in a trial and the way that it looks outside of a trial. And so how do we reconcile that in the real-world evidence space? And especially when we have real-world evidence from a variety of sources. We have EHR, we have insurance claims, we have patient registries. And so today, we'll be exploring how Cardinal Health in our Real World Data and Evidence group, how we've solved for and created some best practices around some of these real-world evidence challenges. At the end of the webinar, we hope and we want you to understand our iterative process, what chart-based data is, how it can be used, and then we hope to bring some clarity to what method you may need for collecting real-world data, how your research needs are being met, and then what that means for everyone from a researcher to a healthcare provider and a decision maker. So with that, we have an excellent panel today. We have an excellent panel of experts who will be providing valuable insights. We hope to make it interactive. We're going to have more of a conversation than just talking at you today. So we encourage you to engage with us. Please submit some of your questions throughout the session into the Q&A panel. And at the end, we'll address some of the questions. So I'd like to introduce some of our panelists. Firstly, it's my pleasure to introduce Dr. Bruce Feinberg, who will be providing a physician's perspective on real-world evidence and the workflow. He currently serves as the Vice President of Clinical Affairs and is the Chief Medical Officer at Cardinal Health. Nationally recognized for his expertise in specialty oncology and the business of specialty healthcare, Dr. Feinberg has been instrumental in the development of clinical pathways that aim to control costs, improve quality, and increase predictability, all of which are key factors that we know in developing sustainable approach for caring for patients for high-cost diseases. Secondly, we have Andy Klink, who is the Senior Director of Business Development in the Real World Data and Evidence group at Cardinal. Andy will be leading the second session on how to select the right patient. He has over 15 years of experience in academia and industry in HEOR, and currently he serves as strategic scientific oversight and business development to support Cardinal's industry-sponsored research studies and involves many types of data, everything from administrative claims, chart reviews, patient physician surveys, and electronic health records. Lastly, but not least, we have Harlan Hayes, and he will be leading Section 3 on interpreting the data. He is a Senior Director of Scientific Operations and Research Analytics in the Real World Data and Evidence group at Cardinal as well. He directs a team of senior statisticians and data scientists in the conduct of statistical analyses and database management across many real-world data sources, from medical chart studies to administrative claims databases, electronic medical record databases, and other sources. So with that, let's get started. I'd like to bring Bruce onto the screen to discuss our first section. So welcome, Bruce.
Hey, Liz.
Let's first kick it off. Talk to us a little bit about the physician's perspective and how Cardinal Health and the group was started.
So I came to Cardinal Health in 2010 And my very first meeting was kind of an awakening into the corporate world of medicine, which I hadn't been in before. I'd been in academic and community practice. And I was surprised by all this talk about TA in this meeting. And I went up to one of the members of the committee that I was asked to be on. And I said, I don't understand why all this interest in temporal arteritis. It's such a rare disease. And they said, no, that's treatment area. And I was just exposed at that very moment to the fact that they have acronyms for absolutely everything. Even where treatment area to me didn't seem like the kind of terminology that needed an acronym. So just kind of an awakening in terms of my beginnings in 2010. Well, it turns out at that point in time, we didn't have a real world evidence shop. And the real world evidence shop grew out of two different business units that were starting to become the backbone of Cardinal Specialty, which today is Cardinal. But then it was a new division being called Cardinal Specialty. So one was a young group of researchers doing mostly market-based research designed for pharma. And the other group was a group I was leading, and they were managing clinical pathways in which the sponsoring insurance company would ask us to become the honest broker with the specialty provider network, with the intent that we could help convey the insurer's concern about the type of care that was being provided and the network's concern with kind of the rules of the road that they feel are impediments to good care. For me, running that business, I thought that we were actually making some really interesting inroads in quality and cost. But I had the greater opportunity for the first time to have all the plans data. I literally had all the data to do cost effectiveness analysis and quality assessments and resource utilization for the 150 oncologists who were voluntarily participating in the program versus the 150 oncologists in the network who chose not to voluntarily participate. And I thought it was so interesting, plus the fact it was good data. We were saving about 20% and we were actually improving quality of care, and which is a great story unto itself. We don't have time for it today, but I wanted to take it to a national meeting, and Cardinal wasn't familiar at that point with the embargo that is required when you submit data to a peer-reviewed conference. And I wasn't familiar at that point with the white paper, which is basically self-advertising. So fortunately, I had sway. We presented it at ASCO in 2012. The data was pretty compelling, and Cardinal said, do more. Flash forward 3 years later, we finally have a real-world evidence shop. So we're young, we're 10 years old. And if you can imagine to this nascent group, the types of things that were coming to us were the problems that nobody else could solve. And often they were biomarker-linked issues when the biomarker can't be coded for, ultra-orphan diagnoses, and just can't find enough of the patients. And what was interesting to me as a clinician and the couple other clinicians who were involved is that we knew all this data was there. We'd all worked in the office, and we knew that it was there, but it's often hard to find. And in every office is like a one-off in which the way they store data is different. So we thought there may be a solution, but it wouldn't be the traditional solution, as you referenced, using claims or EMR. It might require that the treating physicians abstract the charts, and I'm going to use the broad term chart now, in order to find that information.
Interesting. So for those of you who just joined us, we're chatting with Dr. Bruce Feinberg and about how real world evidence, how we collect it, and we're going to go into how it's a little bit different from clinical trials. So Bruce, talk to us a little bit more about that and the practice of EMR and the data there.
Happy to. So we have a graphic I'm going to bring up just for this one answer. It's my only graphic. I'm being very careful about overwhelming with slides. We're underwhelming with slides. But as we were starting to embark on physicians, and I had done some of this work, I had a very large private practice in oncology with 50 physicians and 40 advanced practice providers. And we had started doing real world evidence for industry because the practice was large enough that we had even though it was a single practice, we had 20 sites of care and across multiple states, and it was enough for pharma sometimes to really get a sense of what's happening out there. And what I realized is that there's that which is structured within the EMR, And in that era, there wasn't a lot of EMR use and not a lot of structured fields, but we were pushing, since we had developed EMR, to get more structured fields, but always fighting our doctors who wanted fewer. So there's that which is structured. There's that which is in the narrative, which is unstructured. There's that which may be in some sort of file, often a PDF. That could be a next-gen sequencing report. It could be an external pathology report. There is also data which is going to be at a hospital level for patients whose care is either split between hospital and office, or aspects of care like the initial surgical work. And there's a hospital EHR, but separate from the hospital EHR is usually an archived imaging system, which predominantly is for radiographic images, but sometimes also pathology. And as you can start to see, there's a lot of different pockets, data sets, one might say, of information. So when we think about just going in and looking at an EMR and using NLP, what are we really getting and how much of that are getting? So what the graphic is showing is we research I'll describe in a minute that we're doing. We often reach out to a network of physicians who do research with us before we entertain a project, to understand if they have the patients needed to meet the accrual goals of the client. And in this particular one, knowing that these patients were going to have multiple providers with multiple sites of care for the management of that particular disease, we were querying how much of that data will be found in these different data sets. And it was pretty remarkable. Specific would be relapse refractory DLBCL, diffuse large B-cell lymphoma. So you're going to have the initial journey starts at the community oncologist practice. And then when they are proven to be relapse refractory, at some point they will be referred either for a hematologic stem cell transplant, or they're going to be referred for CAR T, or they're going to be referred for bispecific antibody. They're going to finish that therapy. They're going to come back to the community oncologist practice. And if you need to know about the PET scans and where they're done, or labs at the different sites and times, what are they going to have to access? So what it does, the bubbles just show you that whether it's charted or uncharted, whether it's in the practice's EMR, the hospital EMR, and it will vary practice to practice. A practice which has been acquired by a large IDN or academic institution may be on the same Epic system, and everything may be at their fingertips within that Epic system. But a practice like mine that was truly independent, working with multiple hospitals, in some cases, we'll be able to access the hospital EMR. In some, there'll be a separate archive system for the images. Our next-gen sequencing, we may have ordered rather than being done reflexively at the institution, so it's going to be in a PDF. And so you get a sense from a broad network how often they're going to have to go after these different aspects of the data from these different data sets to really fully complete the queries that would be needed to understand the full patient journey.
Well, sure, that sounds great, right? Like there's such richness of data, but you're choosing it from all these different places. How would you make it fit for purpose? I mean, that sounds almost impossible.
So if we had known what we know today, if the FDA had fully evolved fit for purpose, if they talked about both relevancy and reliability, we probably would just said, we're done. We can't do this. No one can do this. But being naive, and I could say ignorant, which just sounds better than stupid. We thought that none of these things are insurmountable, and so, the old expression, you had to eat an elephant one bite at a time. We weren't trying to tackle everything at once. We were going after individual things as they popped up. And I think that because of the way that we went after the problem, we were able to start to sequentially solve problems. And some of the problems, very early on just had to do with how do we build the reliability that, is critical to fit for purpose. And so we reached out to this market research group of physicians and queried them, how many of you may be interested in doing electronic data capture for patient charts that may require you going outside of the patient record per se? And we were surprised by the number of physicians who thought it was pretty, sound like a pretty cool thing. They wanted to do research. They thought it could be cool if they could do research at their homes. They could be doing the chart abstraction because they can access their EMR at homes or whatever data set. It's all about a login from a computer. and they could do it after their kids go to bed on a Sunday morning, and they're going to be paid fair market value for their time. Now we had to deal with the legal side from Cardinal. We can't just like make up numbers for fair market value. So you had to have external fair market value assessment, which means we needed to have a beta test before we launched the case report form for any project, so that a group of like physicians could tell us how long this should take, and then base that on, and then tell the physicians what the project. And it was each of these individual issues trying to be solved. And that allowed us to start to build a network, which we called OPEN, the Oncology Provider Extended Network. As we started to get projects outside of oncology, then there was the Rheumatology Provider Extended Network, RPIN, and now we have a half dozen of these, blank pen networks. When we started to get asked by pharma, we need to be able to compare apples to apples as we're looking at our clinical trial work. And we can't use surrogate measures like time to next treatment. We need to be able to use PFS. We need to be able to look at CRs and ORR and progressive disease. And so we started to look at how can we do that. And again, we reached out to members of the network and we asked, how often are your radiologic imaging reports giving you RECIST-based criteria on solid tumors? And it was almost never. How often are they giving you measurements of target lesions and non-target lesions? Almost never. Almost all the reporting that they're getting is narrative in the community. And we asked the question, to what extent do you have access to those images? And are you willing, once again, being paid fair market value, to be able to make those measurements as per RECIST so that we could then calculate? And we created and published on Real World RECIST, which is our nomenclature. And we've now done meta-analyses across multiple studies and disease states to do that. So it's been this evolution of trying to tackle and create, the solutions one at a time. And suddenly, you're halfway through the elephant and you're still going strong.
Very nice. So it sounds like you're figuring all this out. So then you think about like what inherent value can this routine care provide in advancing evidence-based medicine? It looks so different from a clinical trial.
So with that work we were doing, again, being somewhat naive, it's like you're feeling like, well, we've done it. We've accomplished it. And once again, it's our clients who we owe so much to, because they keep telling us what more we need to do. And Real World RECIST turned out to be an advance that was valuable, but it didn't allow us to solve all the problems. Was there still selection bias in the patients who were chosen? How do we have an attrition so we know if the patient needed to be consented for a PRO? Where are doctors who just like to do this after hour abstraction going to be willing to take it into the office and add time to their day? Would they be comfortable, again, back in the office environment, having to learn new software to send us de-identified digital image so there's verification of that radiologic work or the pathology? And we realized that to do that, we're asking a lot more. Maybe that's more than our individual physicians and OPEN could manage. So we created the parallel network, PRN. In the practice research network, we're working with practices that do have full research teams who are familiar and used to doing all this work. And so if we do have a project in which we need to get primary source verification, they're used to using that software, doing that de-identified digital transfer. And what it led to is, based on one of the studies for which our data did go to the FDA, it led to work with the FDA, who were curious if we could validate with one additional step and then do BICR. And doing blinded independent central review on real world data really had never been done.
That's amazing. Routine care is so different from randomized controlled trials. We know that. And even if you succeed in, doing this real world RECIST and BICR, how do you overcome these foundational differences?
I mean, there are some things you just can't overcome. Now, you may not be able to overcome them in terms of perfect data matching, but that's where you get in, and Harlan will speak to this, and Andy to some degree, where you've got to develop methodologic approaches. And that has to do with matching, in particularly and sensitivity analyses where you can start to overcome. But there's some basic facts. Physicians don't assess patients at the frequently they're assessed in a clinical trial.
Yeah.
So one of the ultimate goals we thought of this work, and particularly we believe was part of the impetus for the FDA, although never expressly stated, was, we're trying to move from that, from the benchmark of 12 years from the laboratory bench to the bedside, which has been historical for 3 decades. And how is that going to be done? So you can accelerate approvals, and that's one way to get there. And then you're going to have a lot more research after approval, and that's led to some concerns. Another way is to try to shrink that time and cost of phase 3 trials. And the notion had been around forever of historical control, but the feeling always was that, the problem with history is that it's changing, and the antibiotics are better now and the other things, and so it's not apples to apples. But what about patients who were treated concurrently during the exact same time period as this trial population and treated on the standard of care? So we were involved in a project in which the company working with the FDA came to this conclusion that if we could match across these 9 covariates, we believe you could find an external population from real world that could be used as a control arm. So that's pretty intense matching when you're matching across 9. And because our network is so good at patient finding, we were able to be the dominant provider of the 10 to 1 of the 800 stage 2 patients. The external arm was projected to have to be 10 times larger to match across the 9. And of those 800, the majority came from our work. And of that work, we had 80% of the patients in the final cohort that are matched.
Amazing.
And it was interesting of the quality of the data, because it's funny, the things that won't be in the registry, the LDH at the time of index therapy start, like things that you would think as a clinician would always be there. And arbor staging. It was kind of interesting where they were lacking, going to the large national registries or to the major institutional sites that were participating also. So it was that work that led to an FDA review of our data, that led to the FDA wanting to work with us, in which we realize now there are things we can't change. We're not going to change the imaging timing, but there's also an interesting behavioral psychology or behavioral economic bias that's there, because doctors don't want to give bad news. So when you're looking at salvage therapy, the last thing you want to do is tell the patient it didn't work, because they're running out of options. So you're basically telling them they're one step deeper into the grave. And so there is this human element from a treating physician in the community that if the patient's relatively well, it's not like I have something great to offer after this. So let it ride. I don't have to scan them at 8 weeks. They're doing fine. Let's wait till 12. In fact, often you don't start scanning until they're having a side effect and you're forced into it. So that's going to impact how long. If you scan at 8 weeks in this PD, that's a PFS, whereas you don't scan until 12 in this progression, it's 12 weeks. They were progressing at 8 likely, but you didn't look. So in order to address those things, there are the matching and the sensitivity analyses, are mechanistically ways to do it, but it's large populations, but you can get that in real world. And then other forms of sophistication you have to bring to it. But you're now starting from a point where you're 90% aligned, because you're already got BICR. You're already that far down the road. But yes, there are a few steps which will be insurmountable.
Well, thanks, Bruce. I think this is you mentioned selection bias a little bit. You mentioned our network. And so I think it's a good segue to bring Andy Klink on the stage. So thank you, Bruce, for chatting with us. Let's bring up Andy. We'll switch gears into talking about selecting the right patient. So thanks, Andy. Andy, and if you joined late, Andy's an epidemiologist by training. He's currently the Senior Director of Business Development in Real World Data and Evidence at Cardinal Health. So welcome, Andy. Bruce talked a little bit about understanding how the data are derived from real world settings. Let's start talking about how we find the right patient for the study in the first place. So I know there's some important considerations in selecting for patients for real world data. So talk to us a little bit about that.
That's right, Liz. So obviously selecting patients is another key aspect of designing a scientifically sound study, if we have a biased selection of patients, that's likely going to skew the results and give us erroneous conclusions of the effectiveness or safety of a therapy. It could also misrepresent who's actually receiving the therapies in a real world setting. So both of these fall under that theme of selection bias.
So for those of us who know what it is, give us a reminder. And for maybe those of us who don't know what it is, talk to us a little bit about selection bias.
Yeah, so selection bias occurs when we have a set of information that's not representative of the data that we really want to be studying. So there's a mismatch between the sample we have and the target data that we want to understand. So based on that bias sample that we might have, we're going to draw false conclusions. So one way I wanted to depict that is with a well-known painting of called Lincoln by Salvador Dali. And this kind of, if we look at the overall picture, it appears as Abraham Lincoln, and if we zoom in, we see different components within the broader picture. So I'd like to use this to demonstrate how a biased sample of the data, let's say taken only from the woman's hair that's standing on the balcony, might misrepresent the overall picture. So if we were to draw a sample from her hair, come study that, and then draw conclusions of the overall picture, we're likely going to misrepresent the overall picture and have results that don't apply to the broader picture. So really, the theme here is understanding what the target population is. So from there, we can apply the appropriate methods to select the correct sample, but we need to understand what that target population is and what real-world data set contains that target population. So otherwise, that selection bias can lead to results that only apply to that limited real-world data set and not to the broader target population. And that's really due to the way in which the patients were selected into that analytic data set for us. A few examples here, kind of translating it from the painting here to our work is really thinking about, let's say, claims data. So claims might represent only commercially insured patients. So results that we draw from that may only apply to commercially insured patients. Commercially insured patients tend to be younger. It may only represent younger patients. And that claims data set that we have access to might be from a specific insurance plan. So if we're depicting treatment patterns from that data set, we may only be depicting treatment patterns that are unique to that insurance plan.
Yeah. And we know that, selection of patients for a clinical trial can vary differently from how they're selected in a real world study. Maybe let's chat a little bit about that and what the differences are and how the selection differs.
Yeah, so Bruce did a nice job of starting to compare and contrast elements of trials from really how the data are collected. But what I'd like to focus on are kind of two elements of how patients are selected for enrollment and study in a clinical trial. So the first really is the inclusion and exclusion criteria in a clinical trial. So they tend to be very strict and lengthy, and which is really great for minimizing patient variability in terms of characteristics across treatment arms. Great for internal validity of study results, but it may limit generalizability to a broader patient population. And thinking about, again, once that drug is approved, we anticipate that there's going to be a broader eligibility pool of patients that may benefit from that therapy once it's approved. So kind of the trade-off between really high internal validity and limited generalizability may be the case when having very strict selection criteria in a clinical trial. The other aspect I was going to highlight for selecting patients has to do with randomization. If we're looking at a comparison of randomized controlled trial here, randomization of treatment assignment really ensures that balance between both the measured covariates and the unmeasured covariates across treatment arms. If I shift to the real world setting, here's where that real-world data source is going to be influenced with how the data were collected, which Bruce talked about, and where the data are collected. So again, thinking back to that claims example, that might just be from a commercially insured patient population. Another example might be a hospital-based EMR platform, in that the results of that data set that are really patients who have been admitted to a hospital for a certain condition, the results of that data set may only apply to that patient population. So again, applying correct selection criteria after we've identified the right data source is the other consideration. The last thing I was going to touch on quickly on real-world data, a benefit here in understanding that real-world data will likely contain a broader patient population is the ability to look into certain subgroups. So when we think of a trial, that subgroup might be those that match the strict inclusion and exclusion criteria. In a real-world data set, we might think of a subset of interest. We might want to understand how a therapy works in an elderly patient population. So those that are over 70 likely exist in that real-world data set, and allows us to start understanding results that are unique and specific to that subgroup of patients. So if we think about that mural or painting by Dali, again, we may be interested in just the woman on the balcony and not kind of blurring the average with the broader picture.
Yeah, and I think Harlan's going to get into that later, right? About how once we have that, how we look at the data. But talk to us a little bit about, you mentioned about how it's important where we find the data. So where is Cardinal finding patients for real world evidence studies?
Yeah, so that's one of the primary tasks when designing the study is where you're going to find the right patients, right? So again, understanding what that target population is, we have established multiple networks that Bruce alluded to. One is our largest network is our oncology network of oncologists. It's geographically distributed and diverse across the United States. It's primarily community-based practices, which reflects where the majority of cancer care takes place in the United States. It's also agnostic to what EMR platform these practices are using. So we don't have to be concerned with reasons why one practice is using one EMR platform versus another. There may be some kind of channeling bias of why they're using one versus another, as well as other practice affiliations and GPO membership. So we'll have a nice representative sample of patients throughout that network of physicians across the country.
Yeah, the open network is really great and generalizable, right? So let's kind of bring it back. How does the open network are there any methodologies to mitigate selection bias, and what steps has Cardinal taken to kind of operationalize addressing that?
Yeah, so I think one really strong method that we've developed in addressing this upfront is by conducting what we call a feasibility assessment. So on a monthly basis, we're querying our network of physicians to get a sample of patients that we can then start to assess high-level characteristics of that patient sample that we've collected in that assessment, feasibility assessment, and then indirectly compare that to what we know about the target population. So in that sample of patients that we obtained, do those patients look like the target population, what we know of the target population, so that when we do have a study, we know where to go back to, that we have a sample of patients that are representative of that target population that we're interested in. The other aspect that I think is also interesting is that doing these on a monthly basis, we can track potential changes in patient characteristics over time. So when thinking about a newly approved therapy, we may find a sample of patients early on, shortly after approval, characterize those patients, and then 6-12 months down the road, look at a different sample of patients, and they might start looking different compared to what we found shortly after approval. So again, it's always thinking about what is the underlying target population of interest and what that we really ultimately want to study, and then also in the context of some temporal and secular trends of patient characteristics that may change over time.
Yeah, absolutely. Well, I think this is a good time to move into Bill Harlinen. Thanks, Andy. To talk a little bit about, interpreting the data. So we've chatted with Bruce about, the data and how they're derived in a real world setting. And then Andy talked to us a little bit about identifying the right patient. Once we have the patient, Harlan, talk to us a little bit about, are they similar enough to trial patients? Like, what do we do? How do we analyze the data? What do we do with the data?
So in keeping with our little bit of artistic way of thinking about things and stepping a little bit outside of medicine, I want us to think about this as we now actually have way more information than we potentially would in a clinical trial. So in the real world, our goal is to collect anything and everything possible around a potential patient in order to make those decisions later on. Do they in fact match what I'm looking for? So in understanding, do they actually match? I want to step back to why we're trying to do the matching to begin with, right? So if we think of the regulatory guidance we have or have not really received around real world evidence, it starts with the 21st Century Cures Act. It tells us that the FDA in the US would really like us to spend more time looking at data that's already been collected because it's speedier, it's more representative of the general population, but then it didn't say how. And around October 2023, they came up with another document of how to handle the data from a real world regulatory perspective. And it was pretty light in details. And it was really in the, we want the industry to tell us how to do this. So maybe that's a bit of a challenge to everyone that's on the call, is to continue to create ways in which you can highlight what the FDA or the European Union, if you're more on that side of things, is starting to think about this data and actually guide the conversation for them. So in this kind of era, I think it's appropriate for us to say, let's take a leadership role, and we're going to talk a little bit about how I viewed it from a data perspective. But first, let's take a look at, again, the traditional clinical trial. So we're going to match a patient. So we have this data, and I'm going to use music, right? So Liz, I want you to think like the last time that you went to a concert and you looked around the crowd, like how similar was it? Like, did you see lots of different types of people?
Yes.
Yeah, right.
Absolutely.
Especially on the more, I would say, popular things, the more things that are out there. Now you could get hyper specialized. You could get really, really niche in music and in patient selection. And that will help you in the real world evidence side. So let's just look at our matching of a patient, super generic. You have long hair, which is a proxy for phenotype, right? The way something presents. Recently bought a heavy metal album or attended a heavy metal concert, right? So now we have a behavior-based or perhaps environmental exposure into the background. We have this idea that they wear spikes or chains, so that is a choice. So the things that a person has done in their healthcare journey to perhaps lead them here. And then lastly, they have to know the words to Fade to Black. And if anybody knows those words, you're going to know where I'm going with this in a little bit, which is now we're getting into maybe mental acuity or mental status, right? So we create this idea of a person in our vision. And this is who we expected, right? We expected the concert, the rocker, the long hair. By the way, everything I did in here is completely AI generated because it's fun to just see what AI generates in these types of scenarios. So this person has no face, which is actually a good allegory to data. Like we don't know what we're going to get until we actually look at it. And then we don't necessarily understand what the impacts are, right? So we have this, but we already established that everybody that goes to these things that fits these criteria isn't the same. And I would propose that that's what actually makes real world evidence better for regulatory submissions than some clinical trials, is the diversity. It's the aspect that we actually get to see more of what's actually occurring. So if we take our patient here and we apply this criteria, we may get this guy, right? The Southern rocker, the guy who, yeah, he's there. He's got the patches. He's a little bit of a rebel, the long hair. But if you look, it fits every single part of my criteria that you can see. But we may also get the twins, the punk rock rockers. They have the long hair. It didn't say it to be a certain color. So here's already a lesson learned in real world evidence. The better your definitions are, the more concrete it's going to be, right? So I would tell you from a regulatory standpoint, when it comes to documentation, document. It's also going to lead us to a way in which we can do something that Bruce alluded to, which is this concept of a sensitivity analysis. What happens when I throw the punks out and stay more true to just the regular rockers? And then lastly, I just threw this because I thought it was funny. It could be 3 raccoons in a trench coat, because this person literally has just eyes and a giant beard. But you look around it and they're at a country concert. So yes, they kind of fit the criteria, what we talked about, but then they didn't. So it's ours in a data world decision making to actually say, hey, what should and should not be included? So if we translate this back to healthcare, all right, we take our criteria, and you might actually see something more like this, right? You have castrate-resistant prostate cancer. Well, how are we going to define that? Is it by procedural codes? Is it by the other components? Usually by the time it reaches the analyst, there's already been some decisions made. And now we have to live with the repercussions of those decisions of what was included and what was not. So that's why I say, I think we should cast the biggest net possible, right?
Yeah.
So then a choice or a environmental exposure, right? Recently started a PARP inhibitor or another therapy for castrate-resistant prostate cancer. So that is something where now we're seeing action. They failed to respond to radiation. So here's where we start to see the outcomes and impact. And back to my fade to black, they suffer from depression. So here we have the ability to start to build out inside the data what we're actually looking for. So if we take that away, right, and we just kind of start to look at things, now we have this huge database, right? So what do we do with it? What do we do with that information? And I think that's a big part of the conversation is how do we translate raw ingredients and raw materials into something else? Now, I know, Liz, like you've been on the other side of this, right? You've been at pharma, and the ultimate goal, right, is I want patients that look as similar to a clinical trial as possible. And sometimes that data is in there and sometimes that data is not. And so that becomes the challenge that we have within here. So if we have this big data ecosystem, what do we do?
Yeah. So talk to us about that. I mean, obviously these real world data patients look very different from each other. The punk rocker, the twins, the country, lover. Are there certain analytics that are necessary? How do we address this?
So I always think that the best time to address it is before the data was ever collected. And Andy started to hit on this around selection bias and other components. I do think in the real world space, we have something that clinical trials doesn't, which is we actually have more latitude of what gets included. We're dealing in databases frequently. Now, sometimes we're dealing with retrospective or even prospective data collection, right? But a lot of times we're dealing with databases. So it allows us more latitude of being a little bit wider in those definitions up front so we can tighten them up later on. But it really does start with that. If you don't have the right patients in the database, there's nothing you can do analytically to recover from that. So you want to have a good wide view of what's actually occurring out there. If you don't, right, this is where I like to say in real world evidence compared to a clinical trial, you have to get rid of the illusion of control. A clinical trial is all about control. It's all about the ability to say, this is exactly who's in, but not just that, this is the data elements I will collect at very specific time periods. So I'm removing any variation except for the therapy of interest. Like that's the mentality behind it. Now, whether or not you get all the way there, that's a different question. In real world evidence, you have no control over what was collected. As Bruce mentioned, there's different pathways. Certain individuals will or will not collect certain things. So there are inherent biases or decisions made upstream that are going to impact this. So you have to understand that as kind of like step one, right? Is what was collected, what was available. What we're seeing the most common thing for real world is if you do have a clinical trial population, use something like a propensity score match, right? Yeah. To actually bring these patients together and say, it's not just an exact match, which is what we saw with our example, right? Where you can get the exact match and still be wildly different in the actual presentation of the person. A propensity score match, if you're not familiar with it, is based around the idea of the likelihood in observational data that somebody is to receive a therapy and perhaps an outcome. So we're trying to actually say all of this data, this is the advantage of real world data, all of this data combined in here, can we combine this into a more likelihood estimate that kind of says the people that were in the middle, they could have gone to treatment or they could not have gone to treatment. That's the focus point, and those are the real matches. There's going to be some people that no matter what, they were getting this therapy, second line, third line, whatever, because of various things. And there's some people that were never going to get it. It's the people in the middle that the likelihood could have gone either way, that really enables us to kind of say, okay, that's the people I want to match on, because now I've started to, again, bring back some of that control. And so that gives us the ability to do that. Now, to do this, I think that your data has to be large and robust enough to do this. And when I say large, I'm talking about volume, right? It's all about volume, diversity of patients. I don't want to just sample one person in that picture. I want to get the whole crowd as the initial option so that I can understand where people are. Because again, as I get more specialized and have a very specific cancer or rheumatologic condition or reaction or a very specific gastro condition, as I narrow that down, my patient numbers are going to drop down significantly. And yeah, it's going to actually be very close to a clinical trial. But the bigger conditions, they're broader. When I say robust, you want as much data as you possibly can around these individuals. Now, the challenge there goes back to that whole regulatory submission concept, right? Which is the more data I have, the more data I've got to control and be able to source, verify that information.
Yeah.
And get into something that actually, I could trace it all the way back to how was this created? Why was it created? Are there mistakes? Anybody that's played in data long enough, I mean, Liz, I know you've seen it. There's mistakes in EMRs. There are mistakes in charting. There's mistakes in coding. How do you handle that becomes really important for that documentation back to the regulatory component.
Yeah. Maybe talk to us a little bit about how Cardinal does handle that. So how, you talked about clinical trials, the data is meticulously structured and captured, but we want a large representation in real world evidence, how do they capture that, and how do they build out that analytical data set?
Yeah, I think the key that what Cardinal Health has really pushed towards is the ability to have a robust quality control mechanism. And it starts at the data capture level. So a lot of our data is derived through CRF, right, case report form style, so that mimics and feels like a clinical trial. We're allowing the clinician to make the decisions of the definitions. We give them the guidance, but we're relying heavily upon their clinical expertise, as opposed to perhaps maybe a nurse abstractor or an algorithm, right? Something that's out there in the computer world that actually pulls that in. So it starts with that source. And then through there, we're able to look and build out ways to say, does this make sense clinically? Does this make sense from a data perspective? And kind of test back and forth with the source verification to build out something that actually now fits much cleaner, much more robust. So we're taking a lot of the noise out of our concert and bringing it home into something that's much more guided towards what we're looking for in that moment.
Love that. I love that. Thanks for bringing it back to the music example, and I love that we used art examples today too. So I think let's move over to our question and answer. I think we have first. So thanks, Harlan, and thanks to all the panelists today. Really appreciate the time that you spent with us and the great discussion that we had. So I want to first, while we're waiting for some questions to enter the queue, we have a brief polling question for the audience that'll pop up. We'd appreciate it if you answer. And then we'll bring all of our panelists back on for our Q&A. So while we're waiting, let me just start looking at some of the questions that have come through.
So Liz,
I think, yeah, go ahead, Bruce.
I was going to say, while you were picking and choosing and organizing, a quick one that we have on the list is from Tao Ron about our overlap with Flatiron. So I thought I could quickly just knock that one out. So Andy mentioned that our networks are agnostic to their EMR. And so whether you're in US Oncology, One Oncology, whether you're using Onco EMR, which is the EMR, the oncology specific EMR purchased by Flatiron, you could be members of our network. Now we will, with certain clients who will tell us they've already done a report through Flatiron, we will identify the source of data from our participants, but as part of that eligibility and determine that based on that client they don't want overlap, we will not include any Onco EMR investigators within that data pool. So yes, it could overlap. It does. We have means to identify that, and also to, if it's problematic, to deal with it.
Yep. So let's go to another question from Mario. How does an emulated trial study build upon real-world data and real-world evidence?
Who's
Sure,
going
who to kick that over
Yeah,
to?
I
Bruce,
I
you
can
want to take
maybe,
a,
yeah,
or
kick that
Andy?
off, and I'll welcome others to build on that. But I mean, it's certainly building on real world data because it's using observational data, right? So trying to apply RCT criteria to observational data. So there's all of these challenges that have been brought up from Bruce, myself, and others about how endpoints are defined, how selection criteria are defined in observational data may differ from the RCT definition. So while we can apply methodologically the same approach to observational data, there will be a lot of other considerations in target trial emulation, applying it to observational data.
Yeah, I'll just add to that. I mean, if you look back at our kind of 10-year recent history, that is the dominant study that we've done. And essentially, it's because of the fact that historically clinical trials have not been representative. And therefore, there are questions, from a manufacturer regarding patients who are older, patients who are ethnically diverse, better gender representation. And so that is a dominant style. But I also say that it also introduces another issue in a way that Harlan and Andy both address the selection bias and overly rigid eligibility criteria. One of the things that we try to and are aware of now, and you learn as you go, is that how our physicians are going to identify the patients who may meet those eligibility requirements, and which is going to be different when you look back retrospectively at a population and you're going to query your medical record. So if it's highly restrictive, how are you going to try to find that highly restrictive population of this disease that is metastatic to this site? Are you going to start by looking at every patient you've had in the practice with that disease and then going down? Or are you going to first look at those patients who are metastatic? And if you do, does that give you overrepresentation of patients who present with de novo metastases as opposed to those who have recurrent disease with metastases? So I don't want to get too much in the weeds in a Q&A, but we often find ourselves spending a lot of time working with our clients having a lot of dialogue before we ever get close to writing the protocol for the electronic case report form to come to terms around these issues, because they're coming from a world that Harlan described with this incredible rigidity in which you want ultimate control. But if you try to impart that, then the way patients are found and identified often starts to have a lot of selection issues.
And I would say that one of the things I've been floating around is this concept of like a phase 5 trial, right? A phase 4 trial is post-marketing, it's safety and efficacy. Real world evidence, if you're going to emulate a trial, so you're going to take that criteria and apply it here, you also have the ability to look at what I would call treatment adjacent or trial adjacent populations that are part of what I would consider that phase 5. So it gives you the ability to use real world evidence to build upon that emulated trial. We can replicate the trial and then also replicate it in people who were never eligible for the trial to begin with, to give us an idea of what is going to actually happen from not just a safety and outcomes perspective, but getting into the availability, getting into the burden, getting into all these other components in a population that was never intended per se to be studied, because we have them.
I love that. Yeah. Phase 5. Okay, so let's go on to another question that's been posted. So maybe this one's for you, Harlan. With the rise of AI and advanced data analytics, how do you envision the role of real-world evidence evolving in the next 5 to 10 years?
5 to 10 is hard, right? I mean, if we think about what the world looked like 5 to 10 years ago, barely any of us had cell phones at the beginning of that 10 years. By 5 years, we all had cell phones that were smartphones. So I think of it in stages. I think the first place you're going to see a lot of it is we have natural language processing, convert this to this. That already exists. I think the next evolution of it is going to be, give me, as if I were a clinician persona, a definition, and comb through and pull that information out and show me the supporting information so I can void it or confirm it. but it's going to be in sort of the data generation stage. The AI will be utilized in that initial thing. We're nowhere near the trust to just throw data at it and say, what's happening here? And I don't think we're going to be there in 5 years. There's going to be too many regulatory hurdles. There's going to be too much that's blocking it. But I do think in the first 5 years, you're going to see a lot more of these tools taking that unstructured text, taking the radiology, taking a lot of these components and turning them to more usable material. And that'll be the first stage that AI will be utilized. And then later on, we'll start to say, what else did I miss? What did I not see in the data? Tell me something that I didn't already find. And that'll be the next stage.
So I don't know what phase this would be that I'm considering, but it's literally for me almost every day I see study results of a trial in which, by virtue of the timing of the design, the standard arm comparator is no longer the standard of care. And so we have an experimental drug that's looking to get approval, but it's based on a therapy which is no longer the standard. And so how does the treating population look at that when the comparator is not what they currently give? And I think it's one of the greatest impediments to really moving the needle, because as I see that every day, I'm also doing a lot of editing and review work for journals on their manuscripts. And it's one of the issues has always been, why don't physicians follow the evidence? Well, I think we have to address the fact it's really problematic because they don't really know what the evidence is. And you can't cross-journal compare. And so there's got to be a way in real time to be able to give them the evidence they need to really understand where does innovation fit into the daily care of their patients.
Yeah, I think it's that synthesis of new knowledge into something that is usable today. It did remind me of something from my public health days where you would see a sign that you can't swim here today because the E. coli level is too high yesterday. You're like, wait a minute, I was already swimming yesterday, right? It's that idea that there's temporal components, and if it's stale, it's not as relevant. So how do you speed that process up?
Right. It's raining outside and the pollen count's too high, but that was collected in the past 24 hours.
I'm not sure we want to end this webinar on bacteria, but thank you so much, everyone, today. I think that comes to the end of our webinar. I want to thank our speakers for sharing their insights. It's clear that, choosing the right data set, choosing the right patient, choosing the right analyses all play a vital role in advancing and answering research questions for us, improving patient care, and shaping our healthcare decision-making. So thank you again for your engagement, your thoughtful questions that you put in. If we didn't get to your question today, we're happy to answer it at a later date by email, or if you're heading to ISPOR in May, please come and reach out and stop by the booth and say hello. We'll be at booth 1407 at ISPOR this year. So thanks again, and thank you to HealthEconomics.com. I'll pass it back over to Sarah to close us out.
And let me just say, we'll be at ASCO as well.
We will be at ASCO as well.
Thank you all so much for your insights, your wonderful Q&A session. It really was a pleasure to have you with us, and it was a really engaging webinar. And I hope that you all had a little bit of fun too. In closing, we hope that you enjoyed this HealthEconomics.com webinar sponsored by Cardinal Health, and we hope that you enjoy the rest of your day. Thank you.